What is flow cytometry? If we break the term into three parts, flow implies fluid. Cyto means cell, and metry means measurement. So, flow cytometry is a process of performing measurements on cells or particles that are in liquid suspension. It is a technology that simultaneously measures and analyzes multiple characteristics of single cells or particles as they pass through a beam of light. With flow cytometry, researchers can perform many studies, and clinicians can make diagnoses to help treat patients. For example, researchers can study how stem cells mature and differentiate into other cell types. Clinicians can determine the percentages of CD4 positive T helper cells to monitor progression of HIV/AIDS. Flow cytometry is a powerful technology for characterizing and analyzing particles of interest, such as cells, chromosomes, bacteria, and beads. All cytometer samples must be in liquid suspension. Cells from solid tissue must be disaggregated before analysis. Typically, particles or cells from 0.2 to 50 micrometers in size are suitable for flow cytometric analysis. On some cytometers, larger particles can be analyzed. A cell's or particle's relative size, internal complexity, and fluorescence intensity can be measured by flow cytometry. A flow cytometer is composed of three main systems: fluidics, optics, and electronics. The three subsystems work together to simultaneously measure multiple physical characteristics of single particles as they move in a fluid stream through a beam of light. The Fluidics Subsystem brings the particles of interest to the interrogation point, where they interact with the excitation sources. The Optics Subsystem provides the excitation sources and the components to collect light signals and route them to the appropriate detectors. The Electronics Subsystem converts light signals to equivalent electronic signals. The Fluidics Subsystem transports particles in a fluidic stream to the laser beam for interrogation. The portion of the fluid stream where particles are located is called the sample core. The point at which particles meet the laser beam is the interrogation point. Incident laser light is scattered and fluorescence is emitted as particles pass through the interrogation point. The optics subsystem consists of excitation and collection components. The excitation components include lasers, beam-shaping prisms, and lenses to route the laser beams to the sample core. When particles pass through the laser interrogation points, they scatter light. Any fluorescent molecules present on the particle fluoresce. Appropriately positioned lenses collect scattered and fluorescent light. The collection components steer the scattered and fluorescent light to the appropriate detectors. The detectors in the electronic subsystem convert detected light into proportional electronic signals. Electronic signals are then digitized, further processed, and sent to the computer. Data for each cell or particle is stored in the computer and plotted in various ways for further analysis. Now that you have completed this module, you should be able to define the term flow cytometry, describe the types of samples suitable for a flow cytometer, Explain the properties that a flow cytometer can measure. Identify the three subsystems of a flow cytometer.